Now I'll just go. Okay, so thank you very much. Let me welcome everyone to our July edition of our SIAM webinar on algebraic geometry and its applications. And we're very pleased to have Caroline Uhler here from MIT. And yeah, she's going to talk to us about the mathematics of genome packing and regulation. And so I'm very excited. And yes, there you go, Caroline. And please, everyone, you're welcome, as always, to ask questions during the talk, in the chat and the Q&A, and then I will kindly interrupt Caroline and sneak in the question.
Thank you so much. Yeah, please just ask and and just interrupt me anytime. Okay, so what am I going to do today? So I'm talking about a biological question, and we'll tell you a bit about the mathematical questions that arise when actually studying this. So I wanted to start with actually the biological question that has motivated much of what we have been doing over the years, which is the following one. So, you know, if you look at a human and all of the different cells that a human has, we have all these very different kinds of cell types. We have neurons, we have skin cells, we have lung cells, we have, you know, all these different cells. They look all very different and they actually function very, very differently. But the interesting thing is that in every one of these cells, there is the same genome. Okay, that's the DNA that you get from, you know, half from your mom and half from your dad. So every cell has the same genetic information, but it actually gives rise to all these very, very different cell types. So an interesting question that we've been working on a lot is, how is this actually possible? Okay, so how can you have the same information but give rise to all these very different cell types? And now what is quite exciting in biology nowadays is that you can actually look at cells in many different ways. Single cells, you can image them, you can measure the expression of all of our genes. So humans have 20,000 genes. So that means you get out, you know, you measure for every cell a 20,000 dimensional vector. You can image all kinds of different proteins. So you can really probe these cells in many different ways, and you really get to see how these cell types and how even individual cells are different from each other and what the variability is. Okay, so I'll talk about this one question of, you know, how to go between the packing of the DNA to actually then measuring the gene expression, and how to integrate these very different data modalities. And so there, one geometric question that will come up is just the packing of the DNA itself. So this genetic information, how is that packed inside the cell nucleus? Because that will turn out to be quite important if you actually want to get very different gene expression patterns out for different cell types. And then I'll talk more about a geometric question that is related to a machine learning method that we use quite a bit in order to be able to integrate, you know, say imaging and expression data together in order to get a good impression of what actually a cell is. Right? You need to integrate all of the data possible in order to get the best representation of a particular cell in order to really study its gene regulation. So that's going to be a geometric question about a particular machine learning method, that we actually use quite a bit to analyze these kinds of methods. Okay, so at a high level, this is like what has motivated much of what we've been doing. So we have the DNA. So this is actually how you should think of it. It's a 3 meter long strand. So it's like cut up into different chromosomes. So really 3 meters cut up into 46 pieces, all like, you know, nicely packed. That's how you should think of it. That sits inside the cell nucleus. So every cell has a cell nucleus. That's where the DNA sits in. And so, you know, you should think of it as just like a strand where here you see actually every chromosome. It's not like spaghetti inside the cell nucleus. Everything is really nicely ordered. And so we would like to know something about the packing. What is really becoming clear nowadays is that, you know, if this DNA is packed in different ways, it gives rise to very different gene expression programs, different gene regulation programs. And the intuition is quite easy that, you know, if I put 2 genes close in space, they can both be turned on or turned off at the same time. So you have a protein that comes and can turn them both on, right? But if they're far away from each other, then you will not see them be co-regulated. So the packing matters a lot for the expression. And it's known that things like this are also what goes wrong in many diseases. So that, you know, the packing is kind of off, and therefore this will also lead, and it's actually causal. So you can change, you know, you can change the packing of the DNA in these places, and you can actually induce disease states by doing that. Okay, so the packing of the DNA is really quite important, and so that's the first question that I actually want to talk about. So how do you measure the packing of the DNA? So there have been multiple different kinds of ways of measuring this. I'm going to talk about one particular one, which is known as high C. Okay. So what is high C? So what you do is you take, say I care about skin cells, so or immune cells, and you just take many skin cells together, say 100,000. And what you do is there are ways of just kind of gluing together anything that is close by kind of cross-linking them, and then like, gluing together the open, then cutting up everything and then gluing together the open pieces and then just sequencing. So if I sequence here this strand and I sequence this strand, map it back to the DNA, then I know which pieces were actually close together. Okay, and I do this for all these 100,000 cells together. And what comes out is like a frequency matrix like this, where this frequency matrix, what it tells you is that this position I on the genome was, this often close to this position J on the genome in, out of say 100,000 cells. Okay, so that's how you should read this. So this is a frequency matrix of how often any position on the DNA was close to any other position in the DNA on this whole 3-meter long strand. Okay, so now from this information, I would like to reconstruct the whole packing of the DNA inside the cell nucleus. Okay, so this sounds like a very, simple problem. We all know Euclidean distance problems, so probably that's what you'll be thinking about. So here you have the frequency matrix, here you have, right, the packing, the 3D organization of the genome, and, a standard approach, and has been used a lot, and, there are many papers that have done this, is so, we want to get to distances. So this is a problem all in itself of how do you translate frequencies into distances. You can do maximum likelihood estimation, etc., for what is the right kind of, function that goes from frequencies to distances. But let's not worry about this problem for now. So let's turn these frequencies into distances, and then we just have a Euclidean distance problem that can be solved by an SVP. So what is this Euclidean distance problem? How do you solve it? Well, so you have, right, what we want to do is we have our genome, we have all of the locations, say we do it at the level of genes, for example. So we have our 20,000 genes in the genome. So we have 20,000 points that we would like to estimate in 3D because the nucleus is a 3D object. And usually how we solve this Euclidean distance problems is, instead of doing it in the X's, you do it in the gram matrix, right? Which is just this inner product here, which now would be a 20,000 dimensional rank 3 matrix. And we will solve it in these entries here of the gram matrix. And in fact, it is known, and this is really nice work, that you can exactly reconstruct it if there is no noise using a convex relaxation. So although the convex relaxation although it is a convex relaxation, it will actually provide you with the correct, unique, 3D configuration of these points. So what you do is instead of looking for just a rank 3 matrix, you can actually just minimize the trace of this matrix. So that's the sum of these eigenvalues, right? The L1 norm of these eigenvalues. So we'd expect this to give you something low rank, but in fact, it actually gives you the correct rank 3 matrix out, subject to the constraints, right? That these here correspond to your distances squared. And of course, in the gram matrix, this is all up to rotation and of course also translation. So in order to, you just translate everything to be centered at 0. So you just add in this constraint. And of course, the gram matrix is always positive standard. Okay, so this is a standard, this is standard theory for Euclidean distance geometry problems. Very nice that this convex relaxation actually gives you out without noise the true 3D configuration up to translation and rotation. Okay, so what is then the problem, right? We could just solve it like this, so then we would be done. Okay, so what is actually happening here and what is the real problem is that actually the human genome, and for any eukaryotes, this is in fact the case, the human genome is diploid. So I said at the beginning, you get half from your dad and half of the genome from your mom. Okay, so that means that for every chromosome, you have 2 copies. One comes from your mom and one comes from your dad. And the differences on 2 copies of chromosomes are actually very small. Okay, so these differences between different people, where there can be differences between these chromosomes, are very small. These are just some locations. And in general, high C is not able to tell whether it came, to identify between these 2 chromosomes. Okay, you would have to sequence super, super deep in order to be able to tell the difference between these two chromosomes. So how you should think of it is, really you have this problem here where you have all these chromosomes, but for every point on the DNA, you have another point and you cannot identify between them. Okay. So every point has a second copy of it and they are unidentifiable. So instead of getting the distance between any 2 points, you really get the distance between sums of 4 points. Okay, so you do get this distance here between point I and point J, but the problem is that I have 2 copies of points J and 2 copies of points I, right? So really what I get to measure is this plus this plus this plus this. Okay, so you have really 2 endpoints, right? But I have only distances between n choose 2 points. And so what you would like to do is you would like to figure out the embedding of these 2 endpoints. So how many variables you have is 2n times 3 variables, because I want to find an embedding in 3D. So you have 2n times 3 variables, and you have n choose 2 constraints. So not like in the Euclidean distance problem where you have 2n choose 2 constraints, right? Here we just have n choose 2 constraints because we cannot identify between these 2 copies of every locus inside the human genome. So the question is, can you still identify the whole packing like what you can in the standard Euclidean distance problem? First of all, is it identifiable? So it suggests, right, from just counting constraints, you have n choose 2 constraints, you have 2n times 3 variables. Seems like might be, right? Might be identifiable. And if it is identifiable, then it would be great to have an algorithm that can actually output this packing here. Okay, so that's what I'm going to talk about in the first part of the talk. And this paper, you'll find it on the archive, and I saw Kaye is here. So this is work with Kaye, and I'm actually using some of her slides as well. So she will be very familiar with what I'm showing here. And then, so Kaye was visiting us, and then we have Anastasia Believa, so she was a PhD student in my group, and we have another undergrad student who was a master's student in my group, and also an undergrad in the group. Okay, so this is the work I'm going to talk about, and this is here the archive if you want to see. Okay, so let's actually first write it out mathematically, just so that we're all on the same page. So we'll have these, so we'll look at, for us just to have the notation straight. So x, I will use x for the first copy of a locus, so xi, and then yi will be its corresponding copy of the i's locus, and we cannot distinguish between the x's and the y's. Okay, so xi and yi are these 2 copies, like what I had here. So this xi here and this yi here are 2 copies that we cannot distinguish from each other, and xj and yj are 2 copies that you cannot distinguish from each other. And now if you have distances between locus I and j, well, it has to be the pairs, the sums over all distances between any xi and any x or yj. Okay. So these distances are the ones that are given. So for any ij pair, we're given these sums of 4 distances. And now the question is whether we can actually identify these xi's and xj's, which live in R3 for us. Okay, so that's just mathematically written out. Okay, so the interesting thing, and this is the first result, and this is actually proven using linear algebra, which is quite nice, is that in fact, although, if you just do a standard over, just a quick computation of the number of equations and the number of variables, it seems like maybe it would be identifiable. It actually turns out that these constraints are not enough to identify the 3D configuration. So this Euclidean distance problem is not identifiable. And in particular, it is not identifiable up to, and we have this characterized actually, so it is not identifiable up to a sphere. So what you can identify is, and this is quite interesting, right? So xi and yi are exactly these things that right, these copies that can be permuted, right? Xi and yi. But what you can identify is always the distance between these 2 copies. So in that sense, you can identify the center between, and in fact, not just the distance, you can also identify the center between them. So it means that you can identify them up to a sphere around the center here. Okay, so there are infinitely many configurations that satisfy these kinds of distance constraints. In fact, you can identify up to the sphere around the centers between any of these 2 points. They're known as homolog pairs, these guys that always go together, the one from mom and the one from dad. So that's already quite interesting. So, there have been a lot of methods, right, that just apply this kind of these Euclidean distance geometry constraints or any kind of non-convex method to try to identify these configurations. What this says is you can actually not identify it. So whatever they were getting out, is maybe not something that you may want to trust just because there are infinitely many such different kinds of solutions. Okay, so the question is, can we do anything? So it is not identifiable. Are there many other biological measurements that we could use in order to identify the 3D configuration? Just because this is a super important problem, right? We do need this in order to be able to say something about gene expression and gene regulation. So we don't want to stop here. So negative results are good, but not really what we want, right? So, so here are the additional constraints that we will be using. And here we're making use of actually biological measurements that are just becoming available. So just very recently. So here is one paper, and there are more and more of these measurements that are actually becoming available. So first of all, what you can use is just, you know the DNA, right? I know the order of all of my genes. And you can estimate how densely DNA is packed inside the cell nucleus. So you can kind of estimate, at least for neighboring loci, you can say like, okay, so this can be approximated by a straight line, and maybe that's not too far off. So you can approximate like the distance between neighboring loci by straight lines. So maybe that's good enough, right? To get like some more constraints in, at least neighboring loci, you can add in a constraint. But what will really help you, so this will actually not lead to identifiability. So what will really help you are these kinds of data sets that are becoming available, where you not only measure the contact frequency between 2 loci, but you can actually measure whether 3 loci come together, or 4 loci come together, or 5 loci come together. So these are higher order contact frequencies that are becoming available. So it's like a tensor, right? And you actually get to see that 3 things come together. And there have been many biological settings where this has actually been observed, and that it's important that 3 things come together or 4 things come together. So we need to somehow make this mathematical, of what do we mean by higher order distance constraints. It might be clear what you mean by higher order frequencies, but what would you mean by higher order distance constraints, or how could we turn this into a distance constraint in some way? So we want to do it in some way that, afterwards, at least mathematically, we can actually write it out as a convex optimization problem. So this is how we will define it. And I hope it is kind of intuitive. Okay, so let's say we look at 3 loci. Okay, so I1, I2, I3. And notice again, right, for every one of these, we have a second copy. So we have our J3, we have our YI3, we have our YI1, we have our YI2. Okay, and so now you can choose any 3, right? So for each one, I have another copy. So there will be 8 different ones of these, but like, let me just choose 3 of them. And let me say how I'm going to define a distance. So not contact frequencies, but I need to turn this into distances. So this is how we're going to do it. And it's, very much informed by how you think of like somehow distances in the when you have just 2 pairs. So how do you do something about distances in 3 pairs? Well, they all have to come together. So how we will define it is just, we'll take here the center, right? And what we'll define as the distance is just from the center, you just, take all of these distances here, okay, to the center. And now, of course, you have, all of your different possibilities of building a triangle, because for every one of these, I have 2 different copies. So, I can build all these different triangles, and then I have to take the center there, right? And then I take all of these distances here. And we'll just say that, often it's not the case. It's often the case that only one of the triangles, all of these points come really close together, and all the others will not really add up too much to these frequencies. So we'll say that we'll just take here the minimum, so the smallest triangle, and say that most of the frequency counts are actually just coming from the smallest triangle, and that the largest triangles are really not contributing much to the frequencies. So that's what our motivation is for the minimum here, so that we don't have to look at all of these triangles. Most of them will actually just be too far away so that they don't contribute anything to the frequencies. So that's how we'll define something of like distances, higher order distance constraints. And again, how do you go from frequencies to distances? Well, you'll need some function again, something like, to the minus some kind of constant of how you go from the measured frequencies actually to these higher order distance constraints. Okay, so this is what we're going to use. Now the question is, right, we have higher order distance constraints and we have these kinds of constraints of neighboring loci. Do we now get identifiability? Is this enough to kind of break, what we had seen here, all these, infinitely many solutions that we had actually seen here? And so the nice thing is that this is in fact enough. Okay, so if you have these pairwise distance constraints, the ones that we had at the beginning, so the standard pairwise distance constraints, you also have these distances between neighboring loci, and you have these, and here we're just looking at these tensor constraints. And in fact, you don't need all tensor constraints. For simplicity, you can assume all of them, but really what you need is a tensor constraint that involves the first and the last one on every one of your chromosomes. But okay, go with all of them, but we don't need all of them. Then in fact, you do get, you can in fact identify the 3D configuration of the genome. And we also have a nice convex optimization problem, how you can actually write this out. This is also, of course, was motivated us in terms of how do we actually write out these higher order distance constraints so that it can be solved using a nice convex optimization problem. And if we just look at the results of it, so here you see that, we just generate some data, right? So these would be our chromosomes generated using some Brownian motion model, and you have here 6. I hope 6. Here you don't have one, and one is missing. Well, okay, so you have like 2 copies of each chromosome. You just simulate them, basically your diploid setting, and you try to reconstruct them. Then in fact, what you see here, so in the straight line or is the real one, and then like this dashed line here is the reconstruction, and you see that you can exactly reconstruct it when you don't have any noise. We have all kinds of simulations when you also have noise. And you can apply it to real data because this data of these higher order constraints is actually available. So now we can really get these diploid reconstructions that you were not able to get before. And we are able to recapitulate things that people know from imaging studies. Namely, they know, for example, that chromosomes, the larger chromosomes, they tend to be further outside in the cell nucleus. So if you have the cell nucleus, I had a picture at the beginning, the larger chromosomes tend to be outside, the smaller chromosomes tend to be inside. And also what you also get recapitulated, that's also nice because nothing of this was put into the model, is that the more active part, so you have your whole genome, one actually knows, for example, you can just look at gene expression data and you would know which genes are actually more active. And it's also known that the packing is so that, depending on cell type, this is also clear now, is so that the genes that are more active are actually more in the center. Okay, so if you look at, and these are known as A compartments, so the parts of the genome that are more active, they're known as A compartments, and you see that the regions for this particular cell type that are more active actually come out to be more at the center, and the ones that are less active come out to be more outside of the cell nucleus. So some of these things that were, at least we need to check that, whatever we're getting here is kind of reasonable, right? So that, so now actually for the first time, so just a couple of months ago, for the first time ever, it was made possible to, in some cells, actually get the full packing itself, so that you can really visualize the full packing. This is only for now one cell type and very, very few examples. So hopefully now soon there will actually be real ground truth so that one can at least check it, but it's super, super expensive to get this kind of data. Will never be available in large scale. High-C is very easy to get. So, these kinds of reconstructions will be very important, that you can actually do them in many different cell types and really get the packing for these diploid ones, because in the end, we often care about humans. So we do want to know how to reconstruct the DNA of humans.
I have a question maybe at this point. So how do you actually manage to do this solving for such huge data?
Yeah, so in the end, you have this convex it's very similar to the standard one, to this one, right? So we'll write it out in terms of a gram matrix. We have all of our different constraints. We don't have just this pairwise one. We'll also write out the tensor constraints in terms of the gram matrix. We'll write out the neighboring beat constraints in terms of the gram matrix. They all come out to be nice and linear. And so all you have to do is just solve this convex optimization problem. So you can solve it in CVX, for example, and you can solve quite large ones. Now, yes, you're right. If you want to solve really huge ones, you will need to develop specialized solvers for doing this. We did it at the megabase pair level. So, you just make it so as large as you can just solve it using CVX is what we did in this paper. There are specialized solvers for going higher for this particular optimization problems. Yeah, but it's just a convex optimization problem of this form that we need to solve. So an STP.
There's also right now a question in the Q&A, since I already interrupted you, and Carlos is asking if one can say something about the non-generic configurations, and can you say what they are?
Oh,
and
yeah,
if it's possible that it could happen with non-zero probability and real data.
Yeah, so not with non-zero probability. So there are equality constraints. So that's why you would rule them out with positive probability. Yeah. So they would be like, say some distance between two things is 0, things like that.
I see. Okay, things like this. Okay.
So we would just rule them out. Yep. But they might be interesting. I mean, who knows? Maybe there are biological constraints that actually make sure that these happen. And so you're in this non-genericness setting. So it might be important to at some point look at it. But for now, we just say like, yeah, they should be ruled out and it should be okay. Yep. Great. Okay. So anyways, this was a great time to stop because this is kind of the end of the part on like packing, on finding optimal packings of the DNA when actually, you have a diploid organism. There's still many open questions. You can do this at the single cell level, etc. So there it's like a rigidity kind of question when, either 2 points are, so you actually also have these measurements. So there is single cell Hi-C. So you actually also have these contact, it's not a contact frequency matrix anymore. It's just binary. So either 2 points are together or not together. So in general, you don't see, so it could be right that 2 of these copies, for example, are together, since you can still not distinguish between the diploid ones, but you never get to see this at the single cell level, either one is together or it's not together. So you still don't know if you see a 1 there, you still don't know which ones of these 4 possible points that come together are actually together. And so it's a rigidity kind of question, like how many ones does there need to be in this matrix at the single cell level in order for you, and how do the ones have to be distributed in order for you to actually be able to reconstruct the packing of the full DNA at the single cell level. So the data is available. We have not done anything at the single cell level. It's a completely different problem, right, than distances. That's like somehow a rigidity kind of question. You just have 0 ones. These points come together. The question is, is it enough in order to reconstruct the full packing of the DNA? I think there are a lot of different questions that are still open and would be exciting to actually look at for different kinds of data modalities that are in fact available. Okay, but I will move a bit further and tell you about a completely different geometric question that also arises in particular when we want to integrate and move between packing data. And now packing is often also measured in images, like an image like what you see here, and gene expression data. So these are these 20,000 dimensional vectors that you can measure for each one of your cells. I want to be able to translate between them. And here what we have used a lot and others have used a lot in this field are autoencoders. And so what I want to do is like tell you about a really important geometric question that arises when you use autoencoders. Which I think would be really important to understand better, and which I personally think autoencoders are really amazing, have been super useful in biology, and are really, really interesting mathematical object also to study and to in fact study the geometry of them. So what are these autoencoders? And well, okay, so let me actually start here. So, and I'm saying, for sure you've seen this, like neural networks have been super, super successful in this discriminative modeling setting. So you have, images of dogs and images of cats, they're labeled. Now you build a neural network to be able to tell you, yes, this is a cat or this is a dog, right? So just a classification. So image goes in, out comes a class label. Now, generative modeling is different than discriminative modeling, that you don't need class labels. Okay, so I have like all these images, and that's of course often the case in biology. I don't actually have class labels. I get say you take blood, you have many different cell types in the blood, and you just get images of all these cells. You don't have class labels. Okay, now what you would like to do is find a nice embedding where, this embedding is represented or is informative. meaning, for example, that the cats cluster together and the dogs cluster together and the rabbits cluster together. And it should be maybe a bit more. And you'll see afterwards in the problems why I want it to be more. So it should somehow generalize a bit outside of the cats and the rabbits, etc. So meaning that if I walk from a rabbit here to another rabbit here in this space, and I look at the corresponding image, hopefully it's still a rabbit, okay? But also it should be able to do stuff like this, where, you know, if I'm taking a point that is a rabbit and I move towards catland, well, then hopefully in between I get something that is informative as well. And here I would get out a cabbit, okay? So this here in between a cat and a rabbit should look like a mixture of a cat and a rabbit, which would be here a cabbit. Okay, so that's kind of what I would want it to be, right? It should be generalized across, you know, even if I'm a little bit outside my training examples, this should still be an informative space. Now, autoencoders have been exactly what have been used in order to do this. And so how are autoencoders? So what are autoencoders first of all? So they're a particular kind of neural network, just that the autoencoder doesn't go from images to labels. It goes, if you're training it on images, it goes back to images. Okay, and what it does is it goes through this latent space. So it is a neural network that maps whatever your training images are to some latent space, and then from the latent space back to images. And how it's trained, right? We want to get a latent space. So this would be this embedding here. So we would like to get a latent space that is informative. And so that's why how you train this neural network, all the weights of all these parameters here, is just to be able to reconstruct well. So to be able, so that this is written out here, right? So whatever comes out on this side should look the same as what you put in, so that you're losing as little information as possible by going through this latent space. So that will mean that the latent space is quite informative, namely informative enough for you to be able to reconstruct the training images. Okay, so think of it as PCA. PCA is just like, you know, where this is all linear, right? So that would be a linear embedding, but this is a highly nonlinear embedding that you can get by training these autoencoder, this neural network here. Okay, so what can you do with autoencoders? And I'll show you really nice examples afterwards, but you know, you can do really quite nice things. And this will also ask, you know, lead to the kind of question that will come out afterwards, because we're going to use autoencoders in this way. And so maybe you've seen this, and I'm pretty sure you've seen it. I'm sure you've seen like this nice picture of Yosemite in the winter or in the summer, and then the generated image of Yosemite in the winter. And so that's known as style transfer. So this is often done with GANs, but you can also, GANs are much harder to analyze theoretically. You can also do it with autoencoders. And here I'm having a similar picture of how you should think of it. So I'll train my autoencoders on all kinds of images of people, of faces, okay? And you know, there are some people who never smile in pictures. But you know, there are some people who are happy to smile, like Jose. I see him smiling right now. So, you know, I'm sure I could get like a lot of pictures of people where I see Jose once in a neutral face and once Jose in a smiley face, right? And so, and I'll train my autoencoder. And so then there is this person who never smiles. Kathleen also always smiles. Joachim is not smiling. So let's say I can never get Joachim to smile. So then there will be these people who never smile, and I would like to generate a picture of them smiling. Okay. So let me then take Jose, who is nicely participating here. And I have his picture of not smiling and smiling, and I have him in the latent space. And so this vector here, right, would correspond to adding a smile to him. And so in the latent space, what I'll do is I'll just take this vector and put it up here to Joachim, okay? And here comes the dot, right? I've never observed this dot. So I'll just generate, like here for Cabot, right? I have a dot, and I can just generate a particular image corresponding to it. So I'll just generate a picture corresponding, and sorry, my arrows are not working, but you know, I can actually here, I just generate a picture corresponding to it, and out comes Joachim smiling, okay? So this is done a lot, right? So these generative models can actually be used for style transfer. So obviously, so something like this only works if adding a smile is always aligned in the latent space. Okay? So it's quite surprising that something like this can work, okay? Because it will only work if your latent space is really nicely behaved, in particular when adding a smile actually always corresponds to the same vector, and you can just move around your vector to some new person, and out comes a person smiling. So it's quite nice of a geometric space if this actually works, and it does seem to work in many applications. So there are certainly many geometric questions to be asked about these autoencoders. But let me just show you some applications of what we've done and why I think, you know, these autoencoders are super, super amazing. So for example, what you can do is you can translate between different data modalities. So say you have single cell images and you have single cell gene expression data. And so, I can do this using images and autoencoder. I can also, whatever I put in can be my 20,000 dimensional vectors, which are these gene expression vectors. I can train them to reconstruct. And you can kind of glue together data modalities by just gluing together latent spaces. Okay, so I can have images mapped to a latent space back to images and gene expression mapped to a latent space back to gene expression. And then by just, putting them together, right? So then I can go say here from images to latent space, and then I can take the decoder that maps me back to gene expression. I can now actually hop around between very different data modalities. And what I can then do is go from single cell images to single cell gene expression. And we also validated these kinds of models that they actually really work. Just for you to, just so that, this is maybe not so intuitive, right? Because we don't really know how to think about gene expression data. But you can, for example, do the following. You can just take faces again, and you just say my different modalities are black-haired female, blonde-haired female, brown-haired female, black-haired male, etc. And now you can input, right, a say black-haired female, and you can ask, how would this woman look like were she blonde? So that means I take her, move her to the latent space, then move her to the blonde people, blonde women, and out comes a picture of how I believe she will look like when she's blonde. Note that all of these images are generated, right? They're not real images. In particular, you notice it here that it's very hard to generate, say, for example, Asian blonde-haired people. You see that the pictures come out very not clear because, of course, there have not been many training images of blonde-haired Asians, for example, here. So this makes sense. Right? So that's what you can do with these autoencoders. You can also do the following, and this is where you need this alignment. And this is where the theory will come in. So, okay, so in general, so what you would like to do, we did this for COVID-19, we would like to be able to identify drugs that could be used in a new context for COVID-19. So there are these very big, large-scale drug screens that have been performed on cancer cells. And now you would like to predict, well, what happens, right? Of all these drugs that have been applied, can I predict what will happen when I apply them to SARS-CoV-2 infected lung epithelial cells? And then I would like to, you do this, you figure out all of the effects of these drugs, and you would like to find the drug that kind of pushes you back to the normal state, right? You have SARS-CoV-2 infected lung epithelial cell, you have a standard lung epithelial cell, that would be this vector here, right? And so you would want to find a drug that kind of pushes you back towards being normal. So if you can predict the effect of all drugs, well, then you should be able to predict which ones of these drugs is the most aligned with the negative direction of the disease, and then in this way be able to maybe identify some drugs candidates that could be used against COVID-19. Now, okay, how do we predict the effect of a drug? Well, the drug is just like a smile, right? So, I know what this drug will do on these cancer cell lines. Well, so let me try to predict what this drug will do on this new cell type, which is the SARS-CoV-2 infected lung epithelial cell, and then through that predict what it will actually do, just by moving around this vector. Now, I can tell you this doesn't work in general, and pharma industry has, of course, been using things or trying out something like this. So in particular, for example, AstraZeneca, I know that they have tried this out. And that hasn't worked because usually they use these standard autoencoders. And if you're thinking about PCA, this is how you're probably thinking about autoencoding or finding embeddings. You usually go to a lower dimensional latent space. Okay, and this is how autoencoders have always been used. So it has always been used to find a lower dimensional representation of your data. So your latent space is always lower dimensional than whatever you're starting off with. So if you have gene expression data, it's 20,000 dimensional. You often go to something like 100 or 200 dimensional and then back to your original space. So you want to find some lower dimensional embedding. Now, what we're going to tell you to do instead is to do something crazy. Okay, so I'm going to say like, well, do an over-parameterized autoencoder. So instead, try to embed your data into a higher dimensional latent space than what you started off with. Now, this doesn't make any sense. I mean, well, you might think that it doesn't make any sense. I'll tell you that this is what you should do. And this is, in fact, what leads to these alignments. We don't have a proof why it leads to alignments. We have intuition. We have a proof what are the functions that are actually learned by this. But why it is very unintuitive that you should do this is because you now have enough parameters to actually just learn the identity map. So to just learn, I'm going through I don't have a bottleneck anymore in my function, right? So I have a function that goes from some, say, 1,000 dimensional space to a 1,000 dimensional space. I don't have a bottleneck in the middle, so I have more parameters than whatever I started off with. So I could just learn the identity map, and that's of course completely useless, right? Because I don't need to learn and use an autoencoder in order to learn the identity map. But intuitively, so just why this might actually make sense. So if you want things to be aligned in a latent space, it's much easier to align stuff if you have more dimensions available to you, right? So just intuitively, it's probably a good thing to actually go into a larger dimensional space if you want to have stuff aligned that wasn't aligned before. Okay. And now I see that the time is kind of, okay, so I'll do 5 minutes. All I want to do is like give you the intuition mathematically for what is the function that is actually learned by an autoencoder when you go through this over-parameterized latent space. If you want to see how this is applied and why this actually, that this actually is the right thing to do for drugs, then we have here papers that I have added the references to. But I want to give you the intuition for what actually this over-parameterized autoencoder does. I want to just do this very mathematically quickly. Okay, so here is what we're doing, right? So this is our neural network. So you have your training images or expression vectors, whatever. They live in some space Rk. Okay, and over-parameterized can either be you go through a higher dimensional space in the latent space, or you can also just say you have less training examples than whatever your dimension is. That's just the same, it doesn't matter. Okay, so I'll just, for simplicity of like writing out everything, I'll write it out like this. So I have n training examples, they live in Rk, and n, the number of samples, is smaller than the dimension where I'm starting off at. So the autoencoder, what it does is it learns this. Okay, it tries to minimize, right? This is what you put in. And you try to, and this is what comes out, and you try to have this reconstruction be as good as possible. Okay, so now we're minimizing the reconstruction error in my n training examples. Now the thing is, right, this is over-parameterized, so definitely I can get out zero. I mean, I can get out zero training error. So the question is, what is actually the function that this autoencoder learns? Right? There are many functions, so there are many psis here that can give you zero training error. So there are many solutions. The question is which one is actually the one. And usually when you train an autoencoder, what you do is something like gradient descent, right? So theoretically, we'll be not looking at stochastic gradient descent, which is what you usually use, but actually gradient descent, so that we can get theoretical results. So just to get some intuition, let me just write it out as a very simple problem. So say psi is linear, okay? It's actually, and I just have one training example. So, so then what I'm trying to solve is the following problem, right? A times x minus x, and 2 norm, I can put a square. So I'm just trying to solve this. Over A, say, is an R k times k, right? And x, this is a vector, right, in Rk. So this is what I'm trying to solve, right? I'm trying to find some k times k matrix that maps x to itself. Well, okay, there are many solutions, right? For example, A could be the identity. What I showed you before is the identity is not what is being learned by an autoencoder, but A could also be a rank 1 matrix, or it could be a rank k matrix, right? Can be any ranks in between, so it can be full rank or it can be rank 1. So any of these are actually solutions to this system. So the question is, what is actually the solution that comes out when you just train this using gradient descent? So when you just use gradient descent in order to solve this. Now, usually people, when you train neural networks, you actually initialize the weights as close as possible to 0. So you use gradient descent initialized at 0. Now, it's actually quite easy to show that what happens in this linear setting is actually the rank 1 solution is being learned. Okay, so gradient descent will not learn the identity map. It will actually learn a low rank solution, namely rank 1 solution. Question is what happens in the nonlinear setting. So when you actually use autoencoders, and let's just do this example and use an autoencoder to do it. And you'll see that, so I'm just training it on one training example. Okay, autoencoder, a standard one like unit. I'll train it on one training example. And then you just put in whatever, right, into your autoencoder. You just put in some test example. You want to see what is the function that was learned. So I put in something and see what comes out. And you see that what comes out is the training example, or these nice cute 2 little bunnies here. Okay, so it learned actually a point map. It didn't learn a rank 1 solution because, then it would have like the negative of the rabbit as well. In fact, if you use a linear network, it will also learn the negative of the rabbit. So this is much more extreme. It actually learned the point map onto the training example itself. Okay, so that's interesting. And, it is quite surprising because there are many different solutions that could be learned. In particular, if you use a shallow network, actually something comes out that is, doesn't look like, so this UNet is actually quite deep. If you use a shallow network, you learn something that kind of looks like the identity map, okay, but not quite, and I'll show you that it isn't. And if you use a linear network, you actually learn linear combinations. So you do learn, like, say, the negative of the dog, and you learn any linear combination of these things. So the question is, what is actually the function that is learned by these autoencoders? And in fact, and this is going to be my last slide because I think that gives the whole intuition for it. In fact, what we'll do is, I told you that this is actually not the identity. It looks like it learned the identity, but it isn't. So autoencoders are really amazing because whatever comes out, I can just put it back in, right? So here, this thing came out. You see that it's not quite the same, right, than what went in. So what comes out? So let me just actually, whatever comes out, put it back through the function. So I just iterate the function on whatever comes out. So these two look very, very similar, but in fact, this one here is not the same as what I put in. So I'll just iterate it. You just iterate it many, many times. And actually what you'll see, and this is what we prove, is that in fact, if you iterate it, you'll get out your training example. Okay, so the functions that are learned are in fact attractors, and they only have a single attractive point. For one training examples, we have the full proof. They only have one attractor, and in fact, that's your training example. Okay, so although it looks here like they learned the identity map, they did not. They are super self-regularizing, right? They could, if they wanted to, they have enough parameters to learn the identity map. But if you use gradient descent on these highly nonlinear networks, what you're actually learning is a function that is self-regularizing. And in fact, the deeper you are, right, how we saw it here, if you're super deep, this is so self-regularizing that you just output in one step already the training example. So you're immediately contractive to the training example, the deeper you are in your network. If you have a shallow network, like this example here, you see that you actually need many iterations until you actually lead to your one training example. And, I'll just show you the theorem, but this is exactly what it is. So the theorem just says that, for any of your nonlinearities, there is going to be, and we have a closed form formula for the depth that you need and the width that you need. So that, in fact, your training example becomes an attractor, meaning that, right, if you iterate the map, then whatever you put in, you will actually get out your training example. So these networks are highly contractive at the training examples. They're highly self-regularizing. And so it's quite interesting, right, that they don't learn the identity map, although they could. And this was the intuition for us to actually use these over-parameterized systems also for the drugs. So if you want to get somehow spaces that are aligned, and this is what I said, we don't have a proof for why this is actually happening in the over-parameterized setting. If you want things to be aligned, so our intuition was just, here it's contractive at points. So it makes similar points more similar to each other, right? It contracts them to training examples. Maybe it does the same for lines. Maybe it does the same for lower dimensional manifolds. So maybe the effect of drugs that are similar to each other will be made more similar, and thus this works. And so that was just the intuition for actually using it. But this also leads me to all these open problems, right? So we don't actually know yet why these over-parameterized networks actually have this property that they are aligning, or we don't fully know. We just put up one paper on the archive. If you want to have like some of our ideas of why this is happening in the over-parameterized setting, we have it on the archive. But I think there's still so many questions here on like, why do these over-parameterized networks actually align, effects of drugs that are similar to each other? Why can this be used to move between different cell types? There are all these questions, and here we really know nothing about it, about what are these regions of attraction, so these basins of attraction, right? We know that, the points that are kind of close by will be mapped to this training example, but it actually seems to depend very highly on the nonlinearity that you're using. This will be super important for different applications, right? These basins of attraction of what you want to map to a particular training example will be important for applications. So we really need to understand how this geometry of these basins of attractions actually looks like. And up here is just a problem that I already discussed on like single cell high-speed data that is like also completely open. So with that, I'll end and thank all of the group, without which it would really not be possible to have done all this work. I already mentioned the paper that is on the archive with Kaye, with Anastasia, with Lawrence. And the second work here was in collaboration with Misha Belkin, which I also had some references here, which is this over-parameterized autoencoder paper. And with that, thank you all very much and happy to take any questions as well.
Yeah, thank you. Let's first thank all Caroline for this very nice double talk. Very, very nice. Yay. And so the floor is generally open for questions. So also if you want to ask a question orally, you're now welcome to do that, in the sense that I will unmute you. If you use the raise hand tool, I will unmute people. If you don't want to talk. Yes, if you don't want to do that, you are still welcome to use the Q&A and the chat. So while people maybe type the answers, I have a question. Can you go back to this theorem that you did, the last one?
Sure.
I'm just trying to understand how I can read it. So, so you say what any training example can be made. What does that mean? Does that mean like gradient descent can?
No, it will. So sorry, I should be clear. So depending on the nonlinearity that you're choosing and the initialization, like how close you are to zero, that will define, then you can take any training example, but these two things will define how much depth and width you need in order for the training example to become an attractor.
So this is if you do training with a single data point, a single training thing. Okay.
Exactly. Yep.
I see. And so if you would do this with, I mean, like a natural conjecture would in some sense be if I have many training, well, more, let's say more, some small number of training examples that you can still choose a sufficiently large autoencoder such that they all would come. And
we have so many simulations saying exactly that. We can make networks where we have 500 training examples and we can make all of them be attractors by not even very large networks.
But so there is some theoretical difficulty to prove that, or
you are not able to prove it.
Okay.
Yes, it's a conjecture.
Sounds interesting. It's interesting.
Yes.
Okay.
And this is the hard one, I think. We can prove in every example that all of them are fixed points or attractive fixed point, but just looking at the eigenvalues. Even for these examples, well, we don't know how to prove that there are no other attractive fixed points. We can just, run a grid and just look for everything that we put in. It actually converges to one of our training examples. But here we cannot prove that there are no other attractors, and that would
right. So there are somehow two questions. Right? One question is every training data point becomes an attractor, and another question is do other things also mysteriously become attractors? Right.
Yep.
Okay. Right.
That's a harder one, I think.
I see. But your proof for this theorem, it's kind of nice that it doesn't depend on the nonlinearity, actually.
So the depth will, right? So we just have a closed.
Yes. But I mean that you have proof that works. I mean, I thought, maybe one can prove this if I fix something like a nonlinearity, like ReLU. But it's kind of nice that you have something.
I mean, you do have some conditions, but it actually, any of the nonlinearities that people have used are satisfy these conditions. So they're very weak conditions. So.
Nice. Right, other questions? I don't want to ask all the questions. Let's see. I have a problem scrolling, but I don't see any raised hands at the moment.
I can make advertising. I do have here a slide. So if there is anyone, so we have a new center that I'm co-directing at the Broad Institute, so the Eric and Wendy Schmidt Center. We're always looking for postdocs for the center, also research scientists, in fact, as well. So this is a new endowed center by a really generous gift from Eric and Wendy Schmidt and also the Broad Foundation. And so it's, problems between machine learning and biology, and of course, any kind of mathematics that we all like is of interest to you. We have a whole lot of positions open.
That sounds nice. Also, in the meantime, what people might still come with questions. Do you want to say a few words about the proof idea?
For this?
theory, for this last theory. Yeah.
For this one here?
Yeah.
Yeah, I mean,
I don't exactly see, I don't see how I would approach that in some sense.
yeah, so I mean, it is kind of a mess, but it's really it's more of a brute force approach of like really writing out what is the Jacobian, writing out what are these max eigenvalues, and really trying to just push it through. So this is what the proof. I mean, yeah, I don't think it is like super intuitive. Other than out of this proof comes out what are these suitable conditions, and then out of these suitable conditions, you can. Yeah, you get a formula out, and from there you can actually really figure out that you can infer what is your D and your K. That is.
But the proof is in this paper, yeah.
This is in PNS, yeah.
So this is the paper called Over-parameterized neural networks implement associative memory.
Correct.
Okay.
Yep.
Nice.
Yes.
Okay, so otherwise we could maybe do that. Everybody who wants to be around in the more informal question session that we now promoted to panelists, so we can not only hear you but also actually see you. And then so maybe it's nice if you would now use the raise hand tool for everybody who wants to be promoted to panelists, who wants to stay, and then Jose and I can promote you.